So I'm Natalie Saini. I'm an assistant professor at the Medical University of South Carolina. And the title of the project that was funded is Determining the Somatic Mutation Burden and Its Consequences in Scleroderma Lung Disease.
As much as you can in layman's terms or for a patient to understand why your research project is promising for people who are living with scleroderma.
All right, so scleroderma lung disease is actually associated with very high incidence of cancer. In fact, I think it's amongst the top three reasons for death in scleroderma patients who have lung fibrosis, with pulmonary hypertension and fibrosis being the top two reasons. So, what we wanted to understand was what's that link between lung disease, fibrosis, and actually developing cancer downstream. And what our thoughts on this is that it's been shown before that the DNA at its very basic level gets damaged heavily in patients who have scleroderma. Now, that can be due to too many reasons. Some could be, inherent, what you're born with, genetic factors, or maybe over time, inflammation in your lungs caused new defects to come up. Now, all of the DNA damage, in theory, is supposed to result in accumulation of mutations. And mutations are the problem here because as these mutations will accumulate, it's quite possible that over time you will start to get mutations in genes that are cancer drivers. And once you get those, that can explain why somebody is getting cancer downstream, because cancer drivers are literally genes that are supposed to be there to try and protect your, your tissue from developing cancer. So we wanted to see if these mutations, either across the genome or in cancer driver genes, are elevated in scleroderma patients, and that's pretty much what the grant is about.
So this is not only promising to people living with scleroderma, but this is potentially life-saving.
Yeah, so in our world, what we think is that the more we're able to identify the burden of mutations in them, the better we can tell whether or not somebody is going to develop cancer downstream. And secondly, trying to catch mutations in those cancer driver genes is very helpful because the more mutations in cancer driver genes you have, the higher is the probability of you developing cancer. So we're thinking of developing this into something um, called precision diagnostics for individuals, trying to see who with scleroderma can potentially develop cancer downstream.
When you think about this project, what would you say is a particularly innovative aspect of this project?
So the actual technique of trying to detect mutations in people who don't already have cancer is highly difficult as of right now. So I always describe this as, think of each cell in your tissue. So in a given lung tissue, having its own lineage. So since a person was a zygote, each cell division that made them is its own lineage, right? So each cell in your lung has its own parent, grandparent, and they're barely similar to each other. You've got at best very small clones in there. Now, if you were to take a little piece of this and try and sequence it, you would see nothing. You would simply see what this person was born with, because it's really hard to catch mutations at such a small scale. So, what we have is the ability to actually get a single cell out of there and sequence that. Now, that gives us a very wide view at a very small scale and allows us to look at different cells from different people and see how they've accumulated mutations. Um, this is this would probably be the most innovative technique we've got. The second thing that we can do is we can also look very, very deep into a person's tissue. So again, at the single cell level, using this technique called duplex sequencing, which is extremely error free and having very high accuracy, and in trying to look at mutations is extremely important in trying to address what are the causes of mutations or what is the mutation load in a person.
If you had to sum up in one sentence, what is the most important question you want to answer with this work?
I guess the biggest question we want to try and answer is what's the cause of getting cancer in scleroderma patients? What's the cause of carcinogenesis in scleroderma patients?
So, how does this work follow on from work you've done so far over your career?
So when I was a postdoc over at the National Institute of Environmental Health Sciences, we were amongst the first few people who developed this technique of getting single fibroblasts out of a patient's tissue, growing that clonally, and sequencing that. So back then, what we knew was that cancers have a lot of mutation. That was a known fact at that point. What we also knew was that the mutation load in cancers varies drastically, with some tissues having a lot of mutations and some having very few. But the thing we did not know was how many mutations are there in a healthy person who hasn't developed cancer yet. And that is your baseline. If you don't know what your baseline is, how can you define whether or not cancers have more mutations than healthy people? So we developed this technique using skin cells from people, and we sequenced it. And what we found was that skin in healthy people has a lot of mutations, and the mechanisms that lead to those mutations are the same as what we see in cancers. So using the same technique, we are now looking at scleroderma lung samples and trying to see if before somebody gets cancer, what is the level of mutations in their lungs. So
What got you interested in looking at this in scleroderma?
so I think for me the biggest feature is the fact that Carol is has her lab right here, and the first time I spoke to her and we discussed the disease, I was talking to her about this, and I was saying, if scleroderma sample patients have so much DNA damage in them, then they have to have more mutations. And this is something that people haven't been able to address before because they haven't had the tools to do so. It's easier to sequence cancer genomes where you just take a little piece of tumor and actually sequence it because cancers, even though they're very heterogeneous, have some degree of clonality in them. That's not true for healthy tissues, as I was telling you before, or even disease tissues which have not developed cancer yet. So when we spoke about it, we talked about how it's possible that scleroderma patients have increased mutations. And we did a little pilot and we found that that was true. And that was pretty much the impetus into trying to figure out what these mutations are doing and why these patients are getting such high mutations.
How do you see this work potentially impacting the field of scleroderma?
Patients with scleroderma have very high levels of cancer incidence. Trying to figure out why they're getting cancer is extremely important. Now, if we were to figure out what is the baseline of mutations in a healthy lung tissue, figure out how many mutations go up in scleroderma tissues, and we already have a very good estimate of what is the level of mutations in lung cancers. We are in some ways building up this range of understanding what's normal and what's pathological levels of mutations in these patients. Secondly, trying to understand how these patients are accumulating cancer driver mutations will again allow us to A, figure out what's the cause of mutations, and B, try and figure out how we can alleviate this mutation, and finally figure out whether we can use this as a diagnostic for cancer detection in these patients. Now, when I say what's causing the mutations, one of the things we did find was that a lot of these patients have increased oxidative damage. So it might be a possibility to take better antioxidants to try and alleviate some of that mutation levels in them. So try and remove some of the oxidative damage in them. Similarly, we can find out other causes of mutations. We might be able to build intervention strategies to prevent the buildup of mutations in these patients, and then either stave off getting lung cancer or come up with a better diagnostic technique to try and catch that lung cancer before it becomes a problem.
So, kind of thinking into the future, what do you see yourself working on in 10 years from now?
Well, one of the things that we are always talking about in the lab is can we detect this increase in mutations in patients with scleroderma without having to get something as drastic as a lung biopsy. Lung biopsies are highly invasive and not a technique that patients are going to be, amenable to getting repeatedly. So, how do we get cells before we can from them without having to do something so invasive? And one of the techniques is to get cell-free DNA. Cell-free DNA is basically just DNA floating around in your blood. Um, Usually comes off from cells that are dying out. So, can we get an aliquot of blood from a patient? So, just a small blood draw and sequence the DNA that's there and try and identify whether or not these patients have a very high mutation load or whether or not these patients are predisposed to cancer. So, what we are hoping is that the techniques that we're developing in our lab are going to gradually build towards us being able to use cell free DNA from patients and identify whether or not they have mutations in cancer driver genes that might be problematic for them.
It's kind of wild to think about, but it's when you start to start to imagine that
No,
it's possible. Yeah.
well, we are already using cell-free DNA actually in diagnostics right now.
Mm-hmm.
So the most widespread test for this is the prenatal testing that we do.
Right.
We rarely need to do amniocentesis these days to tell people whether or not a fetus might have Down syndrome
Right.
or X chromosome abnormalities. So we can already do that. If we can do that for fetuses, we can definitely do that to detect cancers downstream as well. We've already known to some extent that patients with IBD develop cancer, and using colon cancer screening can help. So, can we come up with something similar for lung cancer? It's definitely one of the things that we are always talking about in the lab, and we're hoping that we can at some point get there.
What do you think are the most promising research directions in scleroderma today or in lung disease today?
That's a really hard question. So, we come from a field of genome instability. So, for me, the most interesting or the exciting part is trying to figure out what are the mutation levels or the different types of genome instability that might occur in people before they develop cancer. And our work currently is entirely focused on trying to figure out mutations. And that is just single changes across the genome, just small changes. But a lot worse can happen to our DNA. Sometimes entire chromosomes go missing. Sometimes you have big rearrangements. So one chromosome just fuses onto another one, or one fuses onto three other ones. Those are complicated changes to detect. And the fact that people are now detecting these in even healthy individuals is very interesting. So, trying to figure out a very full profile of what's normal levels of genome instability, what's a pathological level, is very exciting for us. Uh, To tie that in with inflammatory diseases like scleroderma and trying to see how people who don't have cancer yet but are predisposed to it, what's going on in them, is that's extremely important in my opinion.
If you had unlimited resources, what problem would you want to solve?
Ah, now unlimited resources makes it very exciting for us. So the study that we're talking about for the proposal that we have, we are planning on sequencing genomes from 20 to 30 different people. That's a very small number of people we are talking about. Even if you're going to think of just people who get scleroderma, you've got racial disparities, gender disparities within that one disease. So if I had unlimited money, I would be sequencing so many more people to get a much better view of what different people within our population are like and what the different levels of mutations in them are like. And in my opinion, it is not just possible, it's highly probable that people from different ethnicities are going to have different types of DNA damage just within their lungs because we are all born with different genetic makeup to some extent. So some people have defects in certain types of DNA repair genes, others don't. And sometimes these DNA repair defects actually track with different populations. So if we are able to tell what is the level of mutations and what are the different causes of mutations in people who are Caucasian or African American or Hispanic and so on and so forth, it would be a much better study and would give us a very, very good uh, view into the different causes of carcinogenesis in these people.
I hope you enjoyed learning more about the important work being supported through our peer-reviewed research grants program. We're incredibly grateful to our team of investigators for the work they do to discover the cause, understand the mechanism, and overcome scleroderma forever. The National Scleroderma Foundation is the leading nonprofit supporter of peer-reviewed research in the country. Since our founding in 1998, the foundation has committed over $30 million to scleroderma research. The need to accelerate the pace of discovery in scleroderma is urgent, and we're proud of our leadership role in advancing scientific innovation. Our program prioritizes scientific merit and provides funding for both early career and established investigators. By leveraging the integrity of the peer review process, we can ensure that we are supporting the most impactful science in the scleroderma research community. And when you invest in the Foundation's research program, you can ensure that your resources are being carefully stewarded every step of the way. Thank you for your support of our research programs, and thank you to our investigators for making a difference in the lives of people living with scleroderma.